had come to open recently, 2015, which he had detailed his ideas and works in photodynamic therapy with nanoparticles in ChemCom 2012 and 2013 and 2014 serially. You are going to listen to his talk, and you will enjoy, and you will have a kind of a feeling of prospects of this new possibility. And we have another expert from Canada, and Professor Mariana Folbari. She was as I heard of. This is the first time for me to meet and greet her. And previously, immunologist turned into nanomedicine specialist. So she has authored the several immunity issues related with nanoparticles, and here in this kind of journal. And she's going to talk about and try to give you the sense of the how to understand the immune response to nanoparticles. And then in vitro diagnostics. The lecturers are mostly from this country. And the clinical translation. Professor Min is Seoul National University, the professor of chemistry. She can talk, her ideas how to use the nanographene was detailed in this famous review journal, Advanced Drug Delivery Review, Raman endoscopy, using fluorescence surface enhanced Raman spectroscopic ducts, which was published last year, both for in vitro and in vivo. And he's going to talk about the endoscopic applications. And also following their talk, we have another great person, and Professor Chung. And great means and young and excellent, and who has published a lot and who has so many ideas. And Professor Seok Chung, he is the best expert in microfluidic devices. And here you can just find it, the details, the 3D construction of the cellular microenvironment, and also here to do this kind of a channel type of microfluidics and devices and microsphere, microfabrication. And his talk is entitled The Commercialization of Nano-Interstice Integrated Microfluidic Devices. We have another the lecturer, the Professor Kun. Professor Kun is an electrical engineer, interestingly. He can talk about the in vitro multiplex bioassay. He published in Microfluidics 2015, and here very beautiful looking pictures. Or he can talk about the antibiotic susceptibility test kit, which is in clinical phase 2 trial, which will be used in the clinical setting soon, and I'm waiting for his. But he's going to talk about here this center one, laser pressure punching and single cell genomics. He has a world top technology, so he promoted this to clinical application, and he's going to try to promote this. And this is still under the surface, and you will enjoy. And reality again. Then Professor János Szévényi from Hungary. He's the best-known expert in CARPA, complementary activation, the related pseudobiology, which will deter and block the clinical use of the promising nanoparticles. And we need to understand this very well. And two MDs are going to follow his talk. And Professor Kim is a thoracic surgeon and actually lung surgeon. He has been involved in developing instrument and apparatus, and as well as the nanoparticles. And they're going to talk about the intraoperative image-guided cancer surgery, which he had published in here. He's a surgeon. And he's a cardiologist from Korea, both from Korea University, and he has been involved in developing the application of the optical coherence imaging. He's also been involved in developing the apparatus as well as the nanoparticles, and you will enjoy it. And the last session will be the future prospects. Professor Go can be explained in several ways, but he's in very brief, is editor-in-chief of this new journal, the Journal of Extracellular Vesicles, and he succeeded in delivering using exosomes, extracellular vesicle exosomes, and he's going to talk about the nanocosmos. And as I just try to propagate the idea that liposome is the artificial surrogate of exosome, and exosome is a kind of inherent natural one. So we have best knowledge regarding how to use the liposome, but finally we came to the era to understand the endogenous lipid bilayer structures and nanocosmos, extracellular vesicles. And because of the absence of Kenneth Dawson, I moved to his slot, and my colleague, Professor Huang, is going to talk about imaging strategy and with his unpublished data. And also Byung-Hee Hong is well known, the chemistry expert. In his Nature and Nature Nanotech paper a while ago, he was the first one to develop the nanographene in large area sheet, and he is still the best expert to publish this kind of growth, the development of the chemistry knowledge, growth dynamics, and gas transport mechanism, which I do not understand, graphene liquid cells. But he's going to talk about, during his lecture, and therapeutic effects of nanographene and his collaborative work with the expert, Parkinson expert in Johns Hopkins, and with his unpublished data. And finally, the next generation, you're going to the long time, the editor-in-chief of a very good journal, and Professor Raj Barlow are going to talk about the communicabilities, the last goal of our field. Why? Because it's so multidisciplinary. So it's going to kindly teach you the status of the communication in nanomedicine. And the course objectives are raise the next generation to prepare themselves for nanomedicine. This is our hope. And finally, in smaller scale exchange program between Asian countries. So far, Korea and China, and so far we don't have any Society for Nanomedicine in Japan or other countries in Asia. And exchange between engineers and these PhDs. And why we chose this room for the summer school, and it's kind of international type of discussion room. And please use your microphone, and if you turn on, and you can just ask every question anytime, if the lecturer allows. And we just learned from the Clinam. They are using the opt-out strategy. Because this is a tutorial session, and all the lectures are going to be recorded and streamed to the YouTube immediately. And if the lecturer sign out, as of myself, sign out to opt out himself or herself from this strategy, then unless they sign up, all those materials. So you have saved time and efforts to come here, but your colleagues back in your lab, your juniors and the junior PhD students or the colleagues are still in your lab while just waiting for the coursework or doing experiments, and they can listen to and see. And this had happened several weeks ago. Applied for the IAEA. IAEA means International Atomic Energy Agency, sponsored and tutorial workshop. attracting 47 from Afghanistan to Yemen, at alphabetical order, 47 participants, and their lectures were online. So this was meant to be the lecture course. Here, let me just take Professor Phang's effort. And this was the introduction of the course, and I'd like to introduce Professor Patrick Hunziker from University of Hospital Basel, Switzerland, and he's going to use 40 minutes to explain the state of nanomedicine and the International Society for Nanomedicine. And also,
thank you very much. Now, Korea has contributed importantly to the field of nanomedicine by the historical person of Dasan, which you know much better than I. And he was not only a theorist, but he also focused on practical learning. He focused on innovations of some machines at the time, and he seems to be still a role model today for his compassion of the plight of the lower classes and for his pursuit of sustainable development. So, a very modern view of science. And what we can say today is that medicine has achieved much. This is an image from my intensive care unit, but here in South Korea things look much more modern even, because you are pioneers and real world masters in technology. So medicine has brought so many machines, so many things to save life, to prolong life, to improve health. On the other side, there are other parts of the world where medicine has less advanced. This is myself 30 years ago doing a cesarean section in eastern Congo. And medicine was done at the lower level in this hospital down there. So we see that the medicine of this century has achieved much. It has recognized the causes of most diseases, and it has had spectacular successes in some acute diseases. At the same time, we are transforming acute disease like myocardial infarction into chronic disease states where patients have to take tablets for life. We have not yet learned how to handle individuality in health and disease. We don't look at disease as a system problem, but rather as an individual defect to be repaired. And we cannot cure many diseases, and many diseases are waiting to be eradicated. At the same time, cost of healthcare is a big problem, not only for us, but also for the southern part of the world. Unfortunately, this is the hospital where I worked a few years ago after the Nyankunde massacre. You will not have heard of this massacre because it's far away. It's in a region where they have civil war since 20 years. And what we can learn from this is that healthcare is not only dominated by technology. There are still people living there. Their souls have the same bullet wounds as their houses. And I would just remind us that there is a global responsibility which goes beyond the highly technologized countries. Those who cannot afford it, those who can afford it least, need it most. Is there a future for Africa in terms of healthcare? We don't know, but certainly it will start in reconciliation and peace as a primary fundament of building up a fair society, of a sustainable economy, of an educational system which will finally help us to apply technology to healthcare. The agenda of the medicine of the future means that we need to progress from the medicine today to knowledge-based medicine which focuses on cure of major diseases. Cure may be defined as treat a disease once, and then you can forget it for the next 10 years. Or eradicate the disease from society means no clinical cases occurring in a society. As a cardiologist and intensive care physician, I hope that we will come to the point where we can eradicate heart attack as defined by clinical cases coming to the hospital. So this will eradicate my two professions. We need to personalize, individualize medicine. We need to understand the system aspects, and at the same time we need to be cost effective and high quality. Why did we miss such important aspects, and how should we progress to the future? The progress in medicine, in my conviction, is a progress in knowing, in knowledge. We know that most of our clinical development in pharmaceutical technology is done by starting with mice experiments. And we know that all lab mice are almost the same. At the same time, it's very clear that all humans are not the same, and this has to do with our 1% difference in our genome, which create a huge variability of individual phenotypes, more human phenotypes than atoms in the universe. So each human is markedly different. We also know that it's not only the genome, which is only a few percent of our DNA, which makes the difference between individuals. It's also additional processes like epigenetics, environment, random processes in development that contribute to these differences. So we need to learn that personalized medicine is not just understanding the genome, but goes much beyond that. We know that there is not a single type of breast cancer, but almost as many types of breast cancers as there are patients. And this means that in the future we need to treat individual diseases. We need to treat diseases in an individual, possibly also as individual diseases for themselves. Each cancer is different. We know that diseases change over time. If you treat a severe fungal infection after 12 hours, you have a good prognosis. If you wait one day, then your prognosis is much worse. And we know the same for cardiovascular and other diseases. So a disease is not the same over time. It changes in phenotype, distribution, expression of targets, response to therapy, prognosis, etc. And we need to take these changes into account. The current paradigm of medicine says, the evidence-based medicine says the strongest evidence for therapeutic interventions is provided by a systematic review of randomized, triple-blind, placebo-controlled trials involving a homogeneous patient population and a homogeneous medical condition. And we know that these do not exist, those homogeneous patients and homogeneous disease conditions. So we have a problem here, which we need to overcome. To base medicine in knowledge and to individualize management of a patient and a disease, we need analytical tools. The analytical tools are brought to us by the omics technology, genomics, proteomics, metabolomics, other broad analytical tools, and most of these omics are enabled by nanotechnology. The second important technology to understand individuality of disease and patients are the imaging technologies, and we see more and more that the nanomaterials play an important role in imaging. And I would also like to mention that computer sciences are indispensable for progress. We know a lot about metabolic charts, and conventional pharmaceutical development consists of taking zooming in some area of this metabolic chart, selecting an enzyme, producing an inhibitor, design this inhibitor, test it in cell culture and small animal models, screen for toxicity, and then go to clinical trial phases. So you see, this is a very pinpointed approach to therapy. If we look at disease, at Schwarzenegger when he was young and healthy, he nevertheless had some knee pain because of training too hard. And when he took a painkiller tablet then we know that this painkiller tablet may have solved his pain in the knee, but at the same time put him at risk of kidney problems, of gastrointestinal ulcers, of hypertension and heart failure. So we see that one of the current problems of medicine is the lack of disease site targeting. When we take a drug, we expose the whole body to side effects, while we only treat a localized disease. Now this is an example of generic toxicity. But in the past few decades we have learned that many pharmaceutical toxicities are not generic only, they are also often patient specific, as you see in the past few examples of failed big drugs like the calcium antagonist Hunziker from Basel, Lipogay, producing unpredictable at the time severe toxicity in remote organs. So also toxicity is an important target for individualizing, personalizing medicine. Location barriers are another important variable between patients. If you have a cancer at the location, at any location, compared to, like in these stages, compared to cancer in multiple locations or cancer in the brain or in the pancreas, makes a huge difference to survival. A localized cancer can be cured in many cases. Brain cancer can be very rarely cured like pancreatic cancer. So we need to learn more about compartments, biological barriers, and we will hear a lot in this course because barriers and location and compartments are very important for nanomaterials. To base medicine on knowledge, we thus need to understand and target disease site. We need to overcome those barriers. We need to predict efficacy and toxicity individually. This also requires suited enabling sciences like the material sciences, like the nanobiophysics, like molecular and functional imaging, new pharmaceutical sciences, and innovative toxicologic methods. Nano is the technology fundament for personalized and knowledge-based medicine. Nanoscience is very broad. The notion of nanoscience probably started with the microscopes with atomic resolution, invented nearby to where I grew up, followed soon by similar microscopes that also enabled to handle individual atoms, then the field of nano-optics and photonics, the very important field of materials, computational nanoscience, nanofluidics, complex nanosystems, all of which I will shortly mention in the next few minutes. We need these technologies in a broad way in medicine. One application is the exploration of anatomy and physiology. A second application is the in vitro diagnostics, the clinical imaging, but also the question of prevention, of therapy, of regenerative medicine, and maybe at some point of synthetic biology in the future. Doing such research often profits from having a broad interdisciplinary input. Here you see my research group. It's not a big group, but it's a very broad group. We have computer scientists, biologists, pharmaceutical scientists, epidemiologists, mathematicians. And one of my first exposure to nanoscience was 16, 17 years ago when we did first experiments using these scanning microscopes that allowed the exploration of matter and of science at the bottom, and applied them after they, Gerd Binnig and Heinrich Rohrer applied them to gold surfaces and to sodium chloride atoms on copper surfaces to build a map of Switzerland, as you see here. We applied them to biological materials, like for looking at collagen fiber in molecular resolution by atomic force microscopy in a physiologic solution. So this ability of doing atomic resolution imaging in a physiologic surrounding, not at deep vacuum, was really very impressive at the time. We also looked at living cells in living arteries imaged by AFM, achieving unprecedented resolution for these living specimens with these microscopes. Nanomaterials are one of the key topics of nanomedicine also in this course. And one application of nanomaterials are the novel surfaces. With nanomaterials you can build functionalized surfaces, surfaces that minimize biointeraction for proteins and cells. You can build surfaces that trigger endothelialization, very important for implants. Also the nanostructured surfaces that maximum tissue ingrowth. You can also have drug releasing surfaces that allow to avoid infection of implants. It's also possible to build super hydrophobic and super hydrophilic surfaces, super slippery, super sticky, self-cleaning surfaces, optically active, optically controllable surfaces, offering new tools for analytic devices, diagnostic devices, for example. Antibacterial devices may contribute to prophylaxis, prevention of bacterial infection. Nanostructured surfaces make also sense in the context of nanomechanics. Here you see the nanomechanical cantilevers that are used to build atomic force microscopes. The special feature of these cantilevers is that you can measure their change in location in space by about 1 angstrom by optical means. So they really allow you to look at surfaces at the ultra high resolution. If you coat such surfaces with a thin layer of a material, then you can observe interaction of biomolecules with this thin layer on the surface with extremely high sensitivity. And we have applied this principle using atomic force microscope cantilevers, functionalized with thin surface layers, to use them as a biosensor in respiratory gases. And what we see is that if a patient expires into this system, then this array of cantilever will bend in a very specific way over time and over amplitude, which allows us to apply machine learning, artificial neural networks, so the brain, to make bedside diagnosis in a short time of disease states in these patients. And in certain disease states we have found that there is a very good sensitivity and specificity of such sensors. Similar sensors have been used to detect proteins at very high sensitivity and nucleic acids. As the next thing come the optical microscopes as an enabling tool for nanomedicine. At school we learned that optical resolution is limited to half the wavelength of light, but in the meantime we have had to learn that this classical finding is not completely true. It's possible to do optical imaging at a much higher resolution, as shown by Paul 20 years ago. So he was a colleague of the guys building the atomic force microscope, and he asked himself the question, can I do the same for light? And he was actually able to do that, to build a microscope which at the tip of an AFM, which was hollow, could measure, make light-based imaging, optical images at the resolution of about 30 nanometers. A decade later, the inventors of the super-resolution optical imaging technologies in the far field developed their techniques which we are using now. This is a confocal microscope, so the best thing we could do until a few years ago in terms of resolution. And they have really revolutionized the capability of optical imaging to detect individual proteins and individual protein complexes, also down to the 20-30 nanometer resolution, which got them the Nobel Prize 2 years ago. If you are doing diagnostics in medicine, then you are about to handling fluids. And that means you need somehow to overcome the dry vacuum state of materials, put these fluids on a chip or on another system which you can read out. Something we have also done a few years ago, reducing micro immunoassays to sizes of 30 micrometer pitch size, which allows us to have these mosaic assays measuring multiple disease parameters in a very small scale. Now, if you have such pilot results in your lab, the key question for medicine is how can you reduce technology and physics to a practical system that can be applied in the clinic. And it more and more comes out that the basic physics may be mastered, but we have not yet fully mastered is how to do the things simple in the end to make them really applicable to real diseases and to real clinical scenarios. So we have seen that the limitations of this pioneer approach is practical. It's the question of the macro-nano interface, of the reagent mixing, of the fluid actuation, of mastering the diffusion aspects, and the complexity of such systems. Don't forget that if you reduce systems, that then diffusion becomes much faster, but at the same time producing turbulence in miniaturized system is practically impossible because all flow at the miniature size is laminar, and that means mixing becomes a real challenge. Also detecting molecules without mixing is difficult because if you think about the concentration, for example, of picomolar solutions, if you want to detect picomolar nucleic acid solutions, then you know that a single molecule will occupy 1600 cubic micrometers, and that means in a miniaturized system of a few micrometer size, there may just be no molecule around to be detected. So you run into a problem from continuous analog detection to detection of individual particles which must be there to be seen. We apply currently to use these technologies in real tough field application. This is my PhD student Maurice Mutronigo, currently trying to get to certain places in eastern Congo using this four-wheel car, but even with a four-wheel car it's very difficult sometimes to overcome holes which are 1 meter deep in the rainy season in the main road across his province. We are currently chasing bilharziosis, schistosoma. These are worms which invade the skin in lakes down there, which lead to liver disease in children and in adults. And this is a war zone for 20 years now. It's a complete black area on the map, and what we have found out is that more than 1/3 of the children in this area have this disease. So he has currently visited 50 health districts, looked at 40 children in each health district. So this is a huge survey which he's doing, and which is really alarming because it shows us the blind gaps we still have in healthcare in the world. For novel therapies, the nanomaterials are very interesting and very important. And we are very happy that Tracy has really done the work in the liposome field, and in him you see that coming to a clinical application also requires sound physico-chemical knowledge, because it's not so easy to make something that finally works. There are other materials which are important, like the polymeric materials, pioneered by Speiser theoretically and Kato practically. The supramolecular assemblies like cryptates leading to the Nobel Prize Tulane. The carbon-based materials like fullerenes, carbon nanotubes, and now the graphene sheets. And the dendrimers pioneered by Tomalia. What you observe if you are going from a molecule to a nanosystem is that there are emergent properties, which means that there are properties that come up which were not there in the molecule. For example, a simple example is if you assemble one of our block copolymers, which are simply hydrophilic, hydrophobic, hydrophilic blocks, then you may end up in micelles, or you may end up in vesicles, or you may end up in tubes, or in sheets, or in even more different scenarios. And this is something that cannot as easily be explained by looking more thoroughly at the molecule. So this aspect of emergent properties is highly fascinating from a kind of philosophical standpoint, but also challenging from a scientific standpoint. And there are many emergent properties of material. One is nanoparticle structure, order, shape. Nanomaterials have different surface effects compared to single molecules and compared to macro or microparticles. There are effects like fluorescence quenching, fluorescence resonance, energy transfer, which occur at the nanoscale. There is the effect of surface plasmons in metallic nanoparticles like gold nanoparticles. We see complement activation not seen by single molecules or by microparticles. And there is also the immunogenicity aspect, because nanoparticles have virus sizes. So our defense system has learned to, or is able to defend against nano-size objects. So this is a very fascinating, rich and important field for research. What is more in nano than it's in the single molecules or in micro. If pharmaceutical industry meets the new material, that means that they progress from small molecular drugs to biotech, which they have already done, producing antibodies, enzymes, DNA, peptides. But now they need to go into small molecular ligands, into nanomaterials like dendrimers, photonic materials, inorganic materials, reactive materials, carbon nanotubes. And more challenging even is the nanosystems. If you combine materials, you may combine not only a single type of molecule, but you may combine multiple types of molecules into a single nano-object, which creates a complex nanosystem which may have additional emergent properties. And that's one of the things we are doing in our research lab, combining additional materials into our polymers to achieve functional reactive nanosystems. So from drug molecules to nano-sized drug aggregates, to targetable transporters, to intelligent nanosystems, making simple things in a composite way more complex. And still, the goal is to become better and better in this goal of recognizing the bad cells and hitting them strongly but selectively to eradicate the cancer where it is, while sparing the healthy tissues. You will hear a lot about targeting later. So here you see an example of a human cancer growing as a xenograft in a mouse. And nanomaterials have proven valuable in targeting cancer in various ways, and that's also our experience, but others will report much more. There is a difference between targeting approaches. You may have passive targeting based on the differential vascular characteristics of tumors. There, one risk is that all your material is in the border layer of the tumor, while it's still difficult to hit the deep layers of a tumor, while it's possible also to use active targeting using receptor target delivery, which is a delicate task. But the goal here is to bury much more of your nanomaterial deep in the cancer, because you know that deep in the cancer there is the potential for relapse even if you treat the outside. You also have the cancer-related stem cells, which are a topic of this course also. And so we want to get better in treating the whole of the cancer and not only the surface. Another aspect of targeting is targeting not only cancer, but targeting a cancer cell by intracellular delivery. We have learned that intracellular delivery strongly enhances anti-cancer effects of certain drugs, because in a normal drug you need to traverse the cell membrane before it has an effect, so you have a gradient from outside to inside. But if you can deposit your nanomaterial and anti-cancer drug inside the cell, it is fully hitting this cancer's biology and maybe more effective. Also, pharmacokinetics, local pharmacodynamics are different. So you can have a short injection, the deposition of your material in a cell or in a tissue, and see a long-term effect by slow drug release from this nanomaterial. It's possible also to target diseases which are not cancer. And I'm still impressed by the findings of the complexity of our body by a T cell which is able to selectively destroy a virus-infected cell by the combination of specific recognition of virus protein, of cell surface proteins, by activating a killing mechanism when this combination of virus and self is detected. And we need to learn to, from this complexity, to also design more clever anti-cancer therapies. This is the topic for functional, intelligent and stimulus-responsive nanomaterials that can react to internal stimuli like pH, like redox, like enzymes, to external, internal stimuli like temperature, or be remote controlled by light, by ultrasound, or other, by radiation, to achieve such effects. Doing photodynamic therapy using light switchable nanosystems is one of the promising approaches, because in particular near-infrared light can traverse a certain depth of tissue, and it's possible to design such systems which are light switched in combination with receptor targeting, achieving very high cell killing capabilities. The challenge with these materials is they are relatively complex, and complexity is a real enemy of clinical translation. So if you're doing such an interesting study, you have to figure out how to simplify your design as much to make it clinically translatable and industrially producible. Here we have a system which consists of a polymer shell with size selective channels and pH sensitive enzymes, which allow you to create a system which is inactive in circulation. After arrival at the target in a lysosome, pH drops, enzyme is activated, and you can feed the system with a prodrug which is then activated in situ to produce a desired effect. The reverse can be done by looking at such switchable nanosystems, to use them as nanosensors. In this case, we would like to understand if a certain tissue has a certain enzyme. In tumors, this means we would like to figure out if a tumor could be treatable by a certain drug, focusing on that enzyme, on that enzymatic pathway. And for that, we can target such a nanosystem to the cancer, can load it with a non-fluorescent prodrug, which is then cleaved by the specific cellular cancer pathway to a fluorescent product as seen in this row of images. So this cancer tissue lights up because it contains this target pathway. Without an internalization, there is no such signal to be detected. You can also use such nanomaterials which are targetable to switch gene expression off by receptor targeting of siRNA, as shown in this thesis of ours. And such systems also look like organelles. They are less than 1 micrometer in size. They have their own compartment and their own enzymatic equipment, so they are somewhat similar to organelles, to biologic organelles. So they can be looked at synthetic organelles, which can be used to expand the metabolic capabilities of a target cell, as we have shown here. The native cell cannot convert some precursors to fluorescent products or some biomolecules to their metabolites, but the expanded cell now can do that. So this is a first step towards synthetic biology, where we can repair genetic defects of cells potentially. Unfortunately, Nadav Ben-Haim is back to Israel and is doing something else than nanomedicine today, but he did an important job there. Polymer nanocarriers can be used for novel imaging strategies. Here we have used it for photoacoustic imaging. The idea is that you first expose a tissue to these nanosystems, then you expose it to a flash of light, and you record the image by ultrasound. This overcomes signal to noise issues. If you're going to nanomedicine research, there are some pitfalls. One of the pitfalls is characterization. If you do a study and you report nice results, but you're not really sure what exactly your material looks like, then you are in troubles also for reproducibility. So you need to have thorough characterization facilities or partners available. The next problem is reproducible synthesis manufacture. And even the successful nanomaterials in the past have encountered manufacture and reproducibility challenges, as you may possibly hear later. Then physiology and pathophysiology, cell and organ pharmacokinetics of nanomaterials are markedly different from small molecules and need to be studied. Toxicity of nanosystems is there. There is potential immune toxicity and complement system activation, which can be understood and I believe can be circumvented by design. What is also important is the interaction with regulatory bodies and the question of clinical translation in smaller and smaller patient groups. And for not least is the question of the business models. If you want to have your development in industry, then you need to find a company, and this company needs to find out how they can make money of it. For the question of scalable production, we are moving to miniaturized methods for our miniaturized system. For example, from batch production of nanosystems, we are moving to microfluidic production of nanosystems, as here, this paper based on single-step preparation of injection-ready medical nanosystems without polymers, which really allowed us to improve reproducibility and narrow down variability of our systems. However, we have found that conventional flow-focusing microfluidics are slow in mixing, as I have already explained, and that's why we are moving to more complex geometries. So Strock and Whitesides have published years ago their fishbone design for microfluidics, but if you look at this fishbone design in detail, we say that there is actually a flow separation in two flow regimes, nearly laminar flow in the upper half and nearly stagnant flow in the lower half. So they show some improvement of mixing, but not really a very good improvement. That's why much more complicated microfluidic designs have been tried, and we are currently reporting about novel 3D microfluidic designs like these 3D helixes that allow very nice mixing in a short distance to produce highly homogeneous nanosystems. This is how they look when assembled and when used for mixing two different materials. So you see that diffusion and mixing distance seems much shorter than what we have seen before. You can use this subtractive 3D micromanufacturing approach, which we have published for other things, for example, to produce programmable microsystems. Here you see injecting of a red fluid into one channel, which goes here and then is programmed to follow this route and goes up here and follows this channel. So this really allows you to go beyond standard planar microfluidic system in a highly configurable approach, rapid prototyping, no clean room, good reproducibility, circular cross-section, 3D manufacture, of multiple crossings in one step with a programmable flow. We need to understand in a mechanistic sense how our materials behave, and I will talk about that later. There are still open questions. Biology, is it an analog or a discrete world? If we look at pH, pH is a chemical term determining how acidic your environment is, but if we look at it at the nanoscale, we see that at the pH of 7.4 you have 20 protons per micrometer, and the endosome has a volume of 1/500th of a micrometer, so the question is not what is the pH in an endosome, but the question is there a proton or is there none? So fundamentally, if you go down in size, biology becomes a discrete world, handling with individual events. Something else we are looking for in the future is the precision. So currently our nanosystems still are variable in size, and it's possible to build atomically accurate nanoparticles like gold-25, gold-38 and gold-102, which have atomic precision. All particles have the same number of atoms, and so we are looking for precision. We know that such nanosystems like gold-25 can be highly sensitive sensors for certain events like redox situation, like chirality. What we are also still missing is the quantum effects. We know that quantum physics takes place at the nanoscale, but currently we are still treating nanosystems like conventional drugs, either soluble drugs or microdrugs, and we are somewhat missing the fact that the sheer neighborhood of two nanoparticles has profound effects mediated by quantum effects at this scale, and so we could exploit these quantum effects much better if we understood better how things work. And something very old, you see the first paper of Albert Einstein, 1905, who told about the fact that the light is probably a particle and not a continuum. We are still missing single photons in medical diagnostics, and that, I believe that it's something where nanomedicine will move and has to move soon. To summarize, nanomedicine is a foundation for the medicine of the future. Nanomedicine is an enabling technology for personalized medicine. It requires a broad interdisciplinary approach. Toxicity exists, is understandable and can be circumvented by design. We should consider the pitfalls from the start of this research, and you should discuss your research regularly with clinicians. There are many dead ends in nanomedicine research which could have been avoided by just figuring out where you really can go and should go in your life. Thank you for your interest.
Thank you. And that's just even though we are behind the time, but we can just decrease the lunch time, which is